Very good morning, my dear friends. Today, our eminent resource person, Dr.P.Manjula Madam Garu, Assistant Professor, Department of Botany, Food and Nutrition, RBVRR Women's College Autonomous, Hyderabad. In previous class, Madam has clearly explained about the cell envelopes, that means cell wall and plasma membrane, has clearly explained. Today, Madam will going to explain about the plasma membrane models and the cell cycle, that means cell division also. Madam is going to start today class. Madam, please welcome, start your session. Madam, please share your screen.
Okay, thank you, sir. Sir, is it visible screen?
Yes, madam. Yes, it's visible.
Okay. So today's topic. So today we will discuss about the models of plasma membrane. So before going to the plasma models, so
Please
what is
click
the
on
composition
slideshow,
of?
madam. If possible, please click on slideshow.
Okay. Sir, is it okay?
Yes, yes, madam.
Okay. So plasma membrane, which encloses the every type of the cell, that is both prokaryotic and eukaryotic cells here. So it is physically separate the cytoplasm from the surrounding cellular environment. So plasma membrane, the main function is, so it is the protection of the cell. Okay, so it is a ultra thin, that is 8 to 10 nanometers, and it is elastic and living and dynamic and selectively permeable barrier. Okay, plasma membrane, it is the selectively permeable membrane, and it controls the entry and exit of the molecules or ions, and also generates difference in ion concentration between the interior and exterior of the cell. So the main function is it is selectively permeable membrane. So the term cell membrane, it was coined by C. Nageli and Kramer in 1855. So in 1895, Charles Overton suggested that plasma membrane is composed of lipids. Okay, so he suggested that plasma membrane, it is made up of lipids. In 1900 to 1920, that is further studies on the plasma membrane states that lipid must be phospholipid. Okay, it is attached with the phosphorus group. So in 1925, that is E. Gotter and Grendel proposed that plasma membrane, it is composed of phospholipid bilayer. So it is two-layered. So plasma membrane consists of three major components, that is lipids, proteins, and carbohydrates. So proteins 52%. That is, for example of this is the example of plasma membrane of the human RBC, which contains proteins 52%, lipids 40%, and carbohydrates 8%. So lipids, here again, lipids mainly 4 major classes of lipids are present. That is, first one is the phospholipids. It is most abundant lipids, and sphingolipids, glycolipids, and sterols. Example here, cholesterol. So all of them are amphipathic molecules and possessing both hydrophobic and hydrophilic domains. So protein, according to their position in the plasma membrane, the proteins fall into two major categories. That is integral proteins and peripheral proteins. Peripheral proteins are extrinsic proteins. So integral proteins, these span the membrane, they are present inside, and peripheral, these are on one side or the other side of the membrane. So peripheral in position, integral, they are present inside. So here these are the different types, intrinsic proteins and extrinsic proteins. So these are the lipids, polar region and non-polar region. Carbohydrates, so they are present as a short, unbranched or branched chains of sugars which are attached to the exterior ectoproteins forming the glycoprotein, and to the polar ends of the phospholipids at the external surface of the plasma membrane forming the glycolipids. So here carbohydrates two types, that is glycoproteins and glycolipids. So here the glycoproteins, they are attached, branched, they are unbranched. So they are present as a short, unbranched or branched chains of sugars attached either to the exterior ectoproteins, they are called as the glycoproteins, or to the polar ends of the phospholipids at the external surface of the plasma membrane to form the glycolipids. So proteins are attached with, that is carbohydrates are attached with the proteins, they are called as the glycoproteins. If they are attached with the lipids, they are called as the glycolipids. So models of plasma membrane. So here mainly 3 models are there. That is sandwich model. So it was proposed by Danieli and Dyson in 1935. So in this model, a lipid bilayer was coated on its either side by side with the proteins. So according to this model, so that is in the lipid bilayer was coated on either side with the proteins. So on either side of the lipids, bilayered lipid is present. So on either side, it is attached with the proteins, coated with the proteins. So mutual attraction between the hydrocarbon chains of the lipids and electrostatic force between the proteins and the heads of the lipid thought to maintain the stability membrane. So they predicted the lipid bilayer to be about 6 nanometers in thickness and each of the protein layer of 1 nanometer thickness, giving a total thickness of 8 nanometers. So lipids 6 on either side 1 nanometer thickness of proteins. So total 9 nanometers according to sandwich model. So this sandwich model which was proposed by Danieli and Dyson. So next one is the unit membrane model. So in 1960, Robertson proposed the unit membrane model. So this hypothesis states that so all cellular membranes have an identical trilaminar structure, that is dark, light, dark. So here this one is the dark, so this one is the light, and here again protein layer it is the dark. So this is the Robertson model. So here he named it as a trilaminar. So here also same, that is on either side proteins are present, in the center lipid layer is present. So another model, it was the fluid mosaic model. So in 1972, S.J. Singer and G.L. Nicholson suggested that widely accepted fluid mosaic model. So according to this model, that is plasma membrane contains a bimolecular layer, both surfaces of which are interrupted by the protein molecules. So proteins which occur in the form of globular molecules and they are dotted about here and there in a mosaic pattern. So they are here bimolecular layer of lipid is present. So on which these protein molecules are arranged scattered mosaic model. Okay. So like this. So this is a totally lipid layer on which so these proteins are arranged in a mosaic model. So the proteins, some proteins are intrinsic proteins and some proteins are extrinsic proteins. So experiments on its viscosity suggest that it is of a fluid which consists rather like the oil, and that there is a considerable sideway movement of the lipid and protein molecules within it. So here this lipid, it is a oily and they are here the movement will taking place. So lipid and protein molecules within it. So on account of its fluidity and mosaic arrangement of the protein molecule. So this model of membrane structure, it is also known as a fluid mosaic model. So here this one is the here. So this is the lipid layer on which the protein molecules are present. So this is about the 3 models. So first one is the sandwich model, which was proposed by Danieli and Dyson. Okay. According to this model, so on either side of the lipid bilayer, proteins are present. On either side, proteins are present on this lipid bilayer. So next model is, that is the unit membrane model. So it was proposed by Robertson. Okay. So it is transparent, that is the lipid bilayer lies which is sandwiched between the proteins. So here cellular trilaminar structure, light, dark, light and dark. So next one is the fluid mosaic model. So it was proposed by Singer and Nicholson. So according to this, here protein molecules which are distributed in and on the lipid bilayer in a mosaic pattern, and a lipid bilayer which is exist relatively in a fluid state. Here the lipid is exist in the lipid state, and these proteins are arranged in and on the lipid bilayer. So the embedded protein molecules float on the lipid bilayer. So some of the protein molecules which penetrate on the lipid bilayer. So this is known as the integral membrane proteins, and they partially penetrate the bilayer. Okay, so based on this, that is intrinsic and extrinsic proteins. So these are the main three models. So next one is the membrane protein. So the so here the different types of proteins are present. So cell recognition protein. So carbohydrate chain of the glycolipid and glycoprotein, which serve as the fingerprint of the cell and identify the type of the cell. Okay, it is a cell for same type of cell or other same type of element or other foreign material. So glycoprotein. So it mainly helps in the recognition of the proteins. So glycoprotein on the extracellular surface serve as the ID tags which recognize the individual and different species and different types of cells. So here some cells they act as a recognition. They recognize the foreign materials and their own material. So next one is the integrins. So the integrins, they provide the strength to the cell membrane. And these integrins which are attached to the cytoskeleton and the extracellular matrix to maintain the cell shape and stabilize the proteins. So next one is the intercellular junction proteins. So these proteins which help the cells to stick together to form the tissues. So it binds cells together through the tight junctions and gap junctions. So these junctions are again two types, that is the tight junctions and gap junctions. So these intercellular junctions mainly helps in the attachment. Okay, so tight junction, they seal cells together into the sheet to provide the impermeable barrier to the cell membrane. So it prevents transfer of water and also the water soluble substances through the cell membrane. Example here, intestinal epithelium, tight junctions, for example. So gap junctions, they provide direct connection between the cells, and there are some sort of channels or pores through the membranes of the cells and across the intercellular spaces. So here example is epithelium cells. So here gap junction, they have some channels to transport the material. But in tight junction, there is it will not allow the transfer of water and also the water soluble substances also. Okay, so next one is the enzymatic membrane protein. So different types of proteins are present in the plasma membrane and their function. So enzymatic membrane proteins, these proteins which attach to the integral membrane proteins are penetrate the peripheral region of the lipid bilayer, and they regulate the protein subunits of many ion channels and transmembrane receptors. So maybe as a peripheral membrane proteins. So example here the G protein, that is guanosine nucleotide binding proteins and certain protein kinases. So they mainly act as a enzymatic, they have the enzymatic activity. So they regulate the involved in the action. So next one is the signal transduction proteins or receptor. So these receptor proteins, they bind hormones and other substances on the outside of the cell membrane, and that binding process triggers a change inside the cell called as a signal transduction. So mainly helps in the recognition and receive the signals. So example, the binding of the insulin to insulin receptors causes the cell to put glucose transport proteins into the membrane. So another one is the transport proteins. So these proteins are responsible for the passive and active transport. So passive transport proteins allow water soluble substances, that is small polar molecules and ions, to pass to the membrane without any energy. So passive transport means without they get in the absence of energy also they can transport the material. So active, so they require energy to transport. The cells expends energy to transport the water soluble substances against their concentration gradient, that is active transport proteins. So next, this is about the cell membrane and the composition and the important proteins, membrane proteins. So mainly cell recognition proteins are present in the membrane. They mainly helps in the recognition of the proteins of the foreign or same type. Integrin proteins, they provide the strength to the cell membrane, and they attach to the cytoskeleton and extracellular matrix to maintain the cell shape and stabilize the proteins. Next, intercellular junction proteins, they help to stick together to form the tissues. tight junctions, they provide impermeable barriers to the cell membrane. So here in this only intercellular junction proteins, that is tight junctions and gap junction. So tight junction, they will not allow any water or water soluble substances. Whereas the gap junctions, they have some sort of channels or pores through which the membranes of the cells and across the intercellular spaces. So next one is the enzymatic membrane proteins. These proteins attach to the integral membrane proteins, and here they regulate the protein subunits of many ion channels and transmembrane receptors. So here the G proteins. Example for this is enzymatic protein G proteins. So next is signal transduction. That is, they take the signals and helps in the reaction. So example, binding of the insulin to the insulin receptors causes the cell to put glucose transport proteins into the membrane. So another one is the transport protein. So the proteins, again here two types, that is passive transport proteins and active transport proteins. So passive transport proteins, they allow the water soluble substances without taking any energy. Whereas the active transport proteins, they expend energy to transport the water soluble substances. So this is about the membrane protein. So this is also important. So next one is the cell division. Okay. So cell division. So cell division, it is a process by which a parent cell divides into two or more daughter cells. So cell division usually occurs as a part of a larger cell cycle. So in the cell division, the cell that is dividing is called